For fun's sake and to update your knowledge, let us have a quiz round for you all. Before that quiz, I just want to discuss some sort of information also in between. So it will be learning process also, it will be fun round also. So let us begin with a cell, because we are discussing cell division. So cell, cell and cell. So we have a cell, and in the cell we have a nucleus, and in the nucleus we have discussed about what special structures. They are chromosomes, and these chromosomes contain what? This double helical super variant, that is DNA. So now you have a fair idea, you have enough information about cell division. So let us discuss what do we have next. So here I have shown interphase S stage. So can you recall what is this S stage of interphase? I hope you are able to recall. Okay, I will explain you. Interphase is the resting stage of the cell where there is no cell division. But during interphase, the cell is metabolically the most active. And the interphase is divided into 3 substages. We have discussed G1 stage, then S stage, and then G2 stage. That S stage is the synthesis stage. So what is synthesized? Can you remember, can you recall? What is synthesized? This molecule. Can you tell me which molecule is this? Okay, when this is synthesized, what happens? We have 2 identical copies of this molecule, right? So again the question is, what is this molecule? This molecule is right, this is original DNA, and then we have 2 identical copies of DNA. Means the DNA is replicated now during which stage? S stage of interphase. Next, we move on to mitotic stages. So this is an animation in which you have to guess which stage is this, which stage is this. So here you don't need to guess, I'll explain you what is this. As you can see, initially in the starting we have the first phase, that is the prophase. You can see the chromosomes, centrioles moving towards poles, spindle fibers, and then they reach at the equator in metaphase, they reach at the poles, during which phase? Anaphase, and then finally karyokinesis occurs, that is telophase. So what is actually happening? You can see all the 4 stages in this cell, and remember these stages go on continuously, right? It's a continuous process, it does not stop anywhere. So next you have 4 stages of mitosis. You all know we have metaphase, we have anaphase, telophase and prophase. And here I am going to show you 4 images. First I have told you that first image is showing the metaphase, in which you can see that at the equatorial line in the middle of the cell, all the chromosomes are present. That is why we call it as metaphase, in between. The cells have the chromosomes in the middle, in the equatorial line of the cell. So chromosomes are lined and the equatorial line. This stage is metaphase. But next you all are going to guess what is this stage. Can you guess? Okay, I can give you some hints. See, we have chromosomes, right? These chromosomes, do you think they are now getting replicated? The centromere has got duplicated, has replicated, and we can see sister chromatids now, and they are just going to move apart. So this stage is which stage? The back stage, means the chromosomes are just preparing to go at the poles. So which stage can it be? Yes, you're right, it is the anaphase stage of mitosis. Next, can you see this image? What is happening in this? The chromosomes have reached at the poles, and the nucleus is just going to divide into 2 daughter nuclei. So which stage it is? It is the telophase stage of mitosis. Next we move on to this image. Obviously you don't have any other option, it is a prophase, in which the chromatin material is getting adjusted into what? Into the chromosomes, right? So next, another question from the metaphase stage. Can you tell me where is the pole of the cell in this image? Right, so where there are centrioles, that is the pole of the cell. And where is the equator of the cell? In case of metaphase, you all know that the chromosomes are aligned at the equatorial line. So where the chromosomes are aligned, that is the equator of the cell. So you are giving very correct answers to all my questions. Let us move on to the next question. we have this image, which is a microscopic image of the metaphase stage of mitosis. So chromosomes are lined at the equator. Where can you see those chromosomes? These are the chromosomes, right? So they are aligned at the equator in case of metaphase stage. And where are the esters? The esters will be at the poles. So can you make out the esters? These are the esters which are at the poles of the cells. Very, very correct. Then where are those spindle fibers? You can just make out, they are not so visible clearly, I know, but the spindle fibers are radiated from what? They are radiated from the centriole position, from the esters, the star-shaped structures. So there are the spindle fibers. Next, you can see this very beautiful image of metaphase stage of mitosis. You can see esters. Again, you can see chromosomes are aligned at the equator, right? So next we move on to anaphase. So this anaphase stage is very clear to you all already, that during this stage what is happening? The chromosomes are going to move towards the poles, because this is the back stage. The chromosomes are going to move back. They will reach the poles. So the spindle fiber is attached to the chromosome, and I have talked about something very important, that is the kinetochore fiber. And this attaches itself to the centromere region of the chromatids during which stage? During the anaphase stage. When the chromosomes are going to move back to the poles of the equator, this kinetochore fiber, which has some special proteins that we have discussed, the kinesin protein, this kinetochore fiber attaches to the centromere region of the chromosome, and this is how this helps the chromosomes to reach at the poles of the cell. This is the centromere region, and this is a complete chromosome. Then we have kinetochore spindle that is attached at the centromere region. Also we have the ester which is radiating rays, and we have polar microtubule also. This is how we have a set of microtubules. We have kinetochore region, we have esters, we have centrioles which are radiating which fibers? Spindle fibers, which help in the attachment of the chromosome on the centromere region, the central region of the chromosome. Next, you can see this image, sister chromatids being separated. Okay, you all know that during this phase we have already replicated chromosomes, and we have sister chromatids which are being separated, and they are going to reach the poles of the cell. So this is how they look like. Then this is an anaphase review. You can see centromeres divide into two. Spindle fibers pull sister chromatids to the opposite poles of the cell, and each pole has equal set of genes. And there further will be some sort of nuclear division, karyokinesis, and then cytoplasmic division. So what the cell looks like this, right? Then you can have a review of mitosis over here. Can you see what is happening? The nucleus has divided, and we have a cell prophase. This is the metaphase stage, then anaphase stage, then telophase stage, and finally cytokinesis of the cell. So now here you are going to name the stages. Again a quiz for you all. Which stage is this? Interphase stage, because this is a resting phase of the cell in which the cell is synthesizing everything. Remember, we call it a resting stage, but this is the most metabolically active stage of the cell. Why it is resting stage? Because you don't see any sort of cell division in this stage. Then this is a prophase. You can see that the centrioles are going to move towards the poles, and they are going to radiate esters, and which are going to radiate what? Spindle fibers. So next stage, just tell me, what is this stage? Remember, the chromosomes are in the middle part. So M for metaphase, M for middle, the equatorial line. This stage is the metaphase stage. Next, anaphase stage, because now the chromosomes are going to move towards the poles, the back stage. Then finally the telophase, where karyokinesis means nuclear division is complete. Can you name this? What is this? This is the equator of the cell. Next, can you name this? Yes, you're right. This is the cleavage furrow which is going to separate the cytoplasm of the cell into two daughter nuclei. So you have to now, you'll see one animation of mitosis and name each stage as you see it. So this is an activity for you which you can see. I know you all know this very well. See, what is this stage? Obviously telophase. Then this is cytokinesis. Now we have two daughter cells. Now this is which stage? This is the prophase stage. Nuclear membrane is disintegrated, centrioles are moving towards poles. This is the metaphase stage because now they have arranged at the equatorial line. Then this is which stage? Telophase stage, because now karyokinesis is complete. So I hope that now you are able to make out with me which stage you are able to see in this animation. So I'll wait, you can see all these stages and recall by yourself. Metaphase, anaphase, and then which stage? Telophase. After that, cytokinesis. Cytoplasm is going to divide into two. Next, this type of cells you see in a microscope when you see onion root tip slides. So can you make out everything? Right. So if I talk about, say, this image, see the chromosomes are moving towards the pole. Which stage is this? This is the anaphase stage. So I hope that you will be able to make out for everything.